I'm Dr. Rajendra study of three trace metals in some fishes of Kali estuary, Karwar, Karnataka, India. Abstract: manganese, chromium and copper concentration in the gill, muscle and whole body samples of four estuarine fishes, namely Mugelcephalus, Silagosciahama, Leonathus, Brevistis and Geomorpha sativa, were determined during the period of July to November 2014. Concentration of these metals in different body parts varied from species to species, and the whole body were in the trace metals in the whole body, where in the order of manganese was more than chromium, was more than copper, whereas in sativa the abundance was in the order of chromium was more than copper than manganese. Significant correlation was observed between concentration of metals in gills and whole body in all species except Cephalus. The trace metals are normal constituents of marine environment, and traces are always found in marine organisms. Although at suitable concentration, some trace metals are essential for enzyme activity. They also form an important group of enzyme inhibitors when normal concentrations are exceeded. In the present investigation, the trace metals, namely manganese, chromium and copper in the gills, muscles and whole body of Mugelcephalus, Silagosciahama, Leonathus, Brevistis and Geomorpha sativa, correlated from Kali estuary, were determined, keeping in view the relative biological importance of the above trace metals and also the food value of these fishes. Methods and methods. These four species of fishes were collected and cleaned in seawater, then taken into the laboratory at a refrigerator and thereafter processed. Total 16 fishes were collected and before dissection they were washed thorough in tap water and thereafter in double distilled water. They were then aseptically dissected using clean dissection tools. Gills and muscle were dissected out and kept in clean washed glasses. They were dried in desiccator first for 10 to 15 days at room temperature and then dried at 65 to 70 degrees centigrade in an oven for 24 hours to remove all the moisture. All known quantity, means 100 milligram of each dried sample was taken in a 100 ml beaker. 80 ml of concentrated nitric acid and 2 ml of perchloric acid was added, then heated on a hot plate to near dryness till up to 1 ml solution remained in the beaker. Then 50 ml of double water was then added into each beaker, filtered by watchman 42 filter paper. For whole body samples, the dried fish were crushed to fineness in mortar and pestle, and well mixed sample was taken. Results and discussion. The concentration of these metals in the whole body of Cephalus, Sciahama, Brevis was in the order of manganese was more than chromium, which was more than copper, and in sativa the order was in the form of chromium, copper and manganese. There was highly significant positive correlation between metal concentration in gills and whole body of Brevis, sativa and Cephalus. This is the table, shows the mean trace metal concentration in gill, muscle, whole body of the four fishes, which is Cephalus, Sciahama, Brevis and sativa. The type of metal and its concentration in fishes depend on the type of its diet. This was recorded by Cross-Ital in 1975. For the average moisture content of these fishes was about 72% in whole body and about 85% in muscle. Hence, on the basis of weight, the concentration of these metals in different body parts, especially in muscles, are within the safe limits. These are the references.